Today, we'll be talking about plant endomembrane systems. The endomembrane system is a network of membranes and organelles in eukaryotic cells that work together to modify, package, and transport lipids and proteins. The plant endomembrane system consists of various membrane-bound organelles, including endoplasmic reticulum, Golgi apparatus, trans-Golgi network, TGN, endosomes, lysosomes, and vacuoles. The endomembrane system is derived from endoplasmic reticulum and flows to the Golgi apparatus, from which lysosomes, but despite their high complexity, these endomembrane compartments are highly ordered and finely regulated. The endoplasmic reticulum is a continuous system of flattened membrane sacs and tubules that is specialized for protein processing and lipid biosynthesis. The endomembrane system is important for the cell's compartmental organization to function independently and properly. First of all, we'll start with the endoplasmic reticulum. The endoplasmic reticulum is a network of membranes and flattened membrane tube-like structures. So these are called cisternae. Inside the membrane, a fluid-filled space is there, which is known as cisternae space or lumen, that is continuous with the nuclear envelope. The endoplasmic reticulum acts as an intracellular circulatory or transporting system. It works closely with Golgi apparatus, ribosomes, mRNA, and tRNA. These small dot-like structures are called ribosomes. When ribosomes are attached to the surface of endoplasmic reticulum are called rough endoplasmic reticulum. When no ribosomes are present on the surface of endoplasmic reticulum are called smooth endoplasmic reticulum. Function of rough endoplasmic reticulum and smooth endoplasmic reticulum. First is, it gives mechanical support to the cytoplasmic structures. Second, exchange of molecules by the process of osmosis, diffusion, and active transport. Then, it contains many enzymes which perform various synthetic and metabolic activities. It provides increased surface for various enzymatic reactions. The major function of rough endoplasmic reticulum is synthesis and packaging of proteins. The smooth endoplasmic reticulum is continuous with the rough endoplasmic reticulum but has no ribosomes. Functions of smooth endoplasmic reticulum include synthesis of carbohydrates, lipids, and steroid hormones, and detoxification of medication and poisons. Next is Golgi apparatus. As material travels through the endomembrane system, it will move from one compartment to the next using vesicles to traffic between compartments. Newly synthesized protein always start their journey at the endoplasmic reticulum. Then they are packaged into vesicles and are sent to the Golgi apparatus. From there, they may take different paths depending on their role and destination. Some proteins will be sent to the endosome and then to the lysosomes, while other will head directly for the cell exterior. Even cargo that has entered the cell via the endocytic pathway sometimes comes as far into the cell as the Golgi before being redirected to another destination. The Golgi complex named after an Italian researcher by the name of the Camillo Golgi in the late 18th. He discovered the Golgi during his exploration of the central nervous system by microscopy. Morphologically, the Golgi complex is composed of flattened membrane enclosed sac called cisternae and associated vesicles. In the Golgi complex, cargo enters at one end, the cis phase, for newly synthesized proteins, and exit at the other end called trans phase of Golgi complex. In plants, Golgi stacks are very small and tumble through the cytoplasm using a combination of cytoplasmic currents and the actin cytoskeleton to help them move through the cell. There can be hundreds of these tiny Golgi stacks in the plant cells. Cisternae of Golgi complex are differentiated into three types: cis cisternae, trans cisternae, and middle cisternae. This is a structure of Golgi complex. This is cis phase, trans phase, vesicles, cisternae, and lumen. Cis cisternae, here this is convex phase of Golgi complex, convex phase, or the immature phase of the Golgi complex. They are consisted of a network of interconnected cisternae called cis-Golgi network or CGN. They are site of protein phosphorylation. Then trans cisternae, this phase is concave phase or concave or mature surface of Golgi complex. From it arise secretory vesicles. These are vesicles which carry the modified or mature proteins. They consist of interconnected network called trans-Golgi network (TGN). They are the site of protein glycosylation. Then middle cisternae, they are located between cis and trans-Golgi cisternae. They are connected with them by transport vesicles. Next is function of Golgi apparatus. First is modification of protein. Golgi complex contain different sets of enzymes that are able to modify the received newly synthesized immature proteins. The processes of protein modification include glycosylation, phosphorylation, and sulfation. Glycosylation is a process of addition of sugar molecule and takes place in the middle and trans cisternae. It results in formation of glycoproteins. Then phosphorylation. It is the process of addition of phosphate groups and occurs in cis cisternae. Sulfation is the process of addition of sulfate group into amino acids of proteins. Next function of Golgi apparatus is processing and packaging of proteins, then sorting of proteins. In plant cells, Golgi apparatus has the additional role of synthesizing polysaccharides, some of which are incorporated into the cell wall. Next is trans-Golgi network, TGN. The trans-Golgi network in plant cells is pivoted organelle in the endomembrane system. This is the structure of TGN. This stacked Golgi cisternae has two tubular reticular structures associated with them. One is associated with cis phase and is varyingly known as cis-Golgi network, CGN, as I already told you. Endoplasmic reticulum, Golgi, intermembrane compartment, GIC, or the intermediate compartment, IC. The other tubular reticular structure is known as trans-Golgi network, TGN, and is thought to be the organelle responsible for sorting secretory pathway proteins to their final destination. Plant cells contain at least two subpopulations of TGN: Golgi-associated TGN, GA TGN, or early TGN, and second is free Golgi independent TGN. Now, just look at this structure. It shows vesicles like clathrin coated vesicle. These are vesicles. So this is clathrin coated vesicles. Then other unknown coated vesicles, and these are the vesicles coated with unknown material, and these are budding from a certain area of TGN. Next is vesicles. Vesicles are small membrane enclosed transport units that can transfer molecules between different compartments. Most vesicles transfer the membrane assembled in the endoplasmic reticulum to the Golgi apparatus, and then from the Golgi apparatus to various locations. There are various types of vesicles, each with a different protein configuration. Most are formed from specific regions of membranes, which is when a when a vesicle buds off from a membrane, it contains specific proteins on its cytosolic surface. Each membrane means a vesicle travels to contain a marker on its cytosolic surface. This marker corresponds with the proteins on the vesicle traveling to the membrane. Once the vesicle finds the membrane, they fuse. There are three well-known types of vesicles. First is they are first is clathrin coated, then COP1 coated, COP2 coated vesicles. Each perform different functions in the cell. For example, clathrin coated vesicles transport substance between the Golgi apparatus and the plasma membrane. COP1 and COP2 coated vesicles are frequently used for transportation between the endoplasmic reticulum and the Golgi apparatus. Next is vacuoles. Vacuoles, like vesicles, are membrane-bound sacs within the cell. They are larger than vesicles, and their specific function varies. In plant cells, vacuoles cover anywhere from 30% to 90% of the total cell volume. Most mature plant cells contain one large central vacuole encompassed by a membrane called the tonoplast. Vacuoles of plant cells act as storage compartments for the nutrients and waste of a cell. The solution that these molecules are stored in it is called cell sap. Pigments that color the cells are sometimes located in the cell sap. Vacuoles can also increase the size of the cell, which elongates as water is added. And they control the turgor pressure, the osmotic pressure that keeps the cell wall from caving in. Next is lysosomes. Lysosomes are organelles that contain hydrolytic enzymes that are used for intracellular digestion. The main functions of lysosomes are to process molecules taken in by the cell and to recycle worn out cell parts. The enzymes inside the lysosomes are acid hydrolysis, which requires an acidic environment for optimal performance. Lysosome provides such an environment by maintaining a pH of 5 inside the organelle. If a lysosome were to rupture, the enzymes released would not be very active because of the cytosol's neutral pH. However, if numerous lysosomes leaked, the cell could be destroyed from the autodigestion. So lysosome carries out intracellular digestion in a process called phagocytes. Phagocyte means Greek word phagen, to eat, right? So by fusing with the vacuoles and releasing their enzyme into the vacuole. Through this process, sugar, amino acids, and other monomers pass into cytosol and become nutrients for the cell. Thanks for watching. If you like this video, make sure to subscribe for more. Thank you.